Good morning, everyone. It's a pleasure to have the opportunity to speak to you today about Ultivue's technology, especially in conjunction with Leica and Indica Labs, whose platforms we use on a regular basis. Ultivue is a reagents-driven company focused on expanding the possibilities for highly multiplex tissue imaging with our ready-to-use products and services. Today, I will tell you about what we offer, how our assay works, and how you can apply our technology to advance your research applications. In the field of immuno-oncology, there's great promise for improving patient outcomes if the scientific research community can understand which patients will respond to a particular course of therapy based on their individual tumor microenvironment. What's been found time and time again is that one marker is not enough to be a meaningful biomarker. The tumor microenvironment is incredibly complex with high heterogeneity, and it is important to be able to characterize not only what cell types are present, but also what cellular interactions are taking place by preserving the spatial morphology of the sample. Characterizing the composition and location of immune cells within and proximal to the tumor has been highlighted as important for patient stratification, as demonstrated by this 2017 study published by Bernie Fox's group. In this study, it was found that understanding the density of PD-L1 positive cells alone was not sufficient to differentiate outcomes, but the proximity of PD-L1 positive cells to CD8 positive cells was indicative of overall patient survival. As you can see in the lower plot, expression of a high number of PD-L1 positive cells within 30 microns of CD8 positive T cells was associated with a lower overall survival. We need more biomarker signatures like this to tease out what factors stimulate anti-tumor immune responses versus those that inhibit or suppress the immune system, thereby promoting tumor growth. To achieve this, Ultivue has developed a technology that, one, supports high multiplexing to profile cell phenotypes while preserving precious samples. Two, supports whole slide on-tissue imaging to provide an unbiased map of cellular interactions. Three, is based on a standard IHC workflow that can be easily implemented in any lab. And four, provides high-quality data that is compatible with quantitative analysis. Our portfolio of products is driven by biology to provide the content users require across biomarker discovery, translational, and clinical research applications. Our Ultimapper portfolio includes a series of predefined kits targeted at addressing key questions in immuno-oncology. Our first Ultimapper kits are available now and include the Ultimapper IO PD-L1 kit, which is directed at revealing immune infiltration and determining if a sample is hot or cold. This panel includes CD8, CD68, PD-L1, cytokeratin, and SOX10 markers to classify immune cells, tumor cells, and PD-L1 expression. Likewise, our Ultimapper IO PD-1 kit enables users to determine whether the immune response has exhausted along the PD-1 axis. This panel includes CD3, CD45RO, PD-1, cytokeratin, and SOX10 markers to profile subsets of T cells and characterize tumor cells. In both kits, cytokeratin and SOX10 are provided as a cocktail and are imaged in the same channel since they can be differentiated spatially. Both kits contain all the reagents necessary to run the Ultimapper assay, including the primary antibodies, amplification reagents, labeling reagents, and associated buffers. These kits come fully optimized with ready-to-use protocols, so no additional optimization is required by the user. The Ultimapper approach provides numerous advantages over other multiplexed immunofluorescence approaches, including high multiplexing in situ, sample preservation, plug-and-play operation, streamlined workflows, and versatile implementation across a range of applications. First and foremost, our approach to immunofluorescence imaging enables many biomarkers to be detected simultaneously on a single slide with whole slide imaging without the need for spectral unmixing, so that the greatest degree of information can be captured for each target and sample. Unlike other multiplexed immunofluorescence platforms, the Ultimapper assay enables a single staining step, which yields an efficient workflow with fast sample to answer time and eliminates the need to optimize staining order, providing a faster and more reliable assay development process. The Ultimapper assay is instrument agnostic and can be implemented directly into standard IHC workflows, leveraging autostainers such as the BondRx for high throughput staining, and imaged on a wide variety of fluorescence tissue scanners, including the Leica Versa.